Thanks a lot. Thanks a lot, particularly for Moritz for this fantastic forum. And does it work like that? Okay. So I really do not want to be the downer of the conference when people are really excited about phenomics and they're talking about actually what we cannot learn from phenomics, which is a really downer. But at the same time, by the end of my talk, I really I hope that I convinced that like thinking about the limitation of phenomics, it probably is a really good idea. So please don't get me wrong. I myself is really excited about phenomics, and all the talk that we heard is absolutely fantastic. But is really this is like a bandwagon? Is it a new bandwagon? This sounds really like phenomics is a new thing. Of course, we can do a lot more things, but of course, this big question is what is it good for, right? That is something that we really have been thinking about in a review paper that I wrote with Moritz, Eric, and Seth, and Arthur a couple of years ago. And there is, by now, a couple of review paper that is really excited about phenomics and really talking about this and that is a really good thing to do, which I generally agree with. But what I really want to do is relatively, simple. Here is my daughter's. And as you can see, one of my daughter, the older one, has a deep cut in her forehead. It was just like a month ago. And for some reason, maybe anyone who has siblings, or you have children who has multiple siblings, could probably relate to that. Typically the older sibling makes much more mistakes, and that actually is the case in my sibling as well. The younger children, Maya, who is happily smiling next to the first, is making much less mistake. And what I really like to do in my talk is actually to learn from the older brother or older sister, older sibling of the omics world, which is of course genomics, and learning a little bit from the history of genomics, and after many years of genetics and how genomics really allow us to learn, and from retrospective knowledge of what they could and couldn't do, I kind of want to make a case that what could we expect for phenomics for us to do and for us not to do. And I think one really interesting and probably quite telling place to start is when actually many years before actually even we know what is the gene, the field genetics was already there, and this is probably arguably one of the earliest case for studying genetics, which here I kind of broadly define as a study of inheritance. And what this guy, Francis Galton, did is essentially to take the height for the parents, take the mean value of that, take the mean value of their parents, their children, and then regress them against each other. His idea is that, well, if you know about the information about the parents and regress that against their children, this relationship, that this strength of this relationship will tell us something about the information that we can gain about the inheritance, right? Currently, this correlation coefficient of this mid-parent offspring regression is known as the broad sense heritability, and it is about 0.4. This is kind of really interesting because the foundation of genetics was so before we actually know what the genes are, and that actually means that how conceptual and theoretical actually thinking is already driving the field before what they actually know about the genome. But of course, after that, there was a huge incentive for geneticists and molecular biologists to of course find out what the actual genes are. And these two books are really great. It's really about the history of genetics and molecular genetics, and they really talk a lot about what happens after the finding of Galton's work. And up to the time of 50s, we all know that Crick and Watson finally found what the genes are, but there was a lot of history what actually people think about and what really people did in terms of finding the gene. And then there seems to be, this is like one of the quote from one of the book that is really, thick, and probably you do not want to buy it, but probably you can get it from the library, and this is in the preface of it. And this quote really says quite a lot. During the rise of molecular genetics in the 50s, which is after the Watson and Crick's finding, any statistical description of inheritance, including Francis Galton's work, became far less appealing, indeed, to many geneticists, quantitative genetics, the statistical genetics, so to speak, appears an anachronism, or like an outdated approach, a clutch that biologists no longer needed. And I think many senior colleagues here probably will really be able to relate to this with your own experience, that according to a lot of writing and historical knowledge of how the genetics as a field work, it seems that the actual sentiment of genetics or molecular genetics in the 60s and 70s appear actually to seem to really feel that they could kill the statistical genetics. Really, there seems to be have a really strong incentive to really do that, and that is a really interesting piece of history, I think. However, this is really cool and interesting part of it. After 124 years, after Francis Galton did this parent offspring regression, there were a group of molecular geneticists who tried to recall the finding of Francis Galton, the 40% of heritability, which is shown in this table, if they use the non-molecular, completely molecular-free method to estimate heritability of human height, it is about 40%. Using the molecular and genomic data, what they could recall is only 3.8% of the heritability, and it was like a sensational result because where are the rest of the 34% or 36% of the heritability went? Where is it? Like if genome is the information, we should be able to explain 40%, but we couldn't, and it basically became this scandal caused the case of missing heritability, and by now there I think is a pretty good example and explanation for how to increase this predictability of heritability, which is essentially sort of include both the molecular data but also the classical statistical genetic information in addition to the molecular data. What it really seems to tell me is that this is simultaneously remarkable and encouraging, because the century-old only pencil and a paper method seems to outperform the revolutionizing genetic methods so much, like an order of magnitude. And to me, this is a really good reminder that there are something to learn from this, and I think this is what I think is the case. The first is that I have no complaints about molecular genetics. It really provides us a great array of methods, right? And it's a PCR, QDLM mapping, and most recently CRISPR. It really allowed a lot of tool to understand how genome work. On the other hand, the understanding that the genomics really provide us relatively little approach for us to better understand why genome works the way it is. The way we want to understand those why questions still is basically based on the classic molecular and population genetic method, even though I'd like to, of course, mention that is not necessarily true, that those two people, Moto Kimura and Tomoko Oda's work on the molecular neutral evolution in the molecular neutral model for molecular evolution is both inspired by the molecular data and also population genetics. So it's not necessarily true that we didn't learn nothing, but by and large, the methodological revolution was what we learned from genomics and much less about the concept and theory. And that is what I think we are facing as well, because this is just like a screenshot of the talk that we have given during this workshop. And of course, which is not a really bad thing, but it's really method-oriented. We really want to use new method to either address old questions or new questions, but there is relatively little about theoretical challenges about studying phenotypic data. So based on those learning from the past, what I would like to propose is that what phenomics will very likely to provide is, first of all, it will increase sample size, that's for sure, which is a great thing. And second of all, I think this is a really good thing that they will give us a consistent measurement error. And I'm not necessarily saying that computer visions are more accurate, but it's the important thing is that they will provide us always a similar type of measurement error. We can measure them many, many times. We can evaluate measurement errors. That is a great thing to me. And also the cost is going to be reduced and new kinds of measurement. And we have already learned a lot about that during this workshop, so I don't really have to delve into what kind of thing we can do with those data. But at the same time, what is quite unlikely for the phenomics itself to do is to solve some of the fundamental conceptual and theoretical issues associated with studying phenotypes. And there are a couple of things, and probably you have many different kinds of ideas, but what I could come up with on top of my head was something like a measurement theory, what it means to measure a thing, and relates these measurements to the meaning of biological entity. This is something that we always have to care about, and it's so difficult in phenotypic data. Actually, even more difficult than genomic data, and that is quite troubling. And the other thing is, character identity, which is a little bit, more jargony. It's things that developmental biologists are always talking about, what actually it is mean for a character to be an independent character, and how do we define that? Or what is a homology? We have discussed a little bit about, comparing, skulls of one species and skulls of other species. That probably is possible, but how can we compare, insect wing with some other, insect that actually doesn't have a wing, like it just doesn't have it, like how do we do that? So like those things, phenomics will really never solve, and this is some kind of thing that probably we need to identify. Is there anything that we can learn for those conceptual issues from the phenomic data? If it is, what is that? And if it is very difficult for the phenomics to solve those issues, then we should be quite humble that the limitation of phenomics is still there, and we should bear with those limitations. So with that in mind, I really go very quickly. I don't really want to go much deeper into what I do, but I'm working again with Eric Svensson in this department, but also in collaboration with Seth. And with Seth and also the data that I collected in collaboration with Eric, we have been basically building a machine learning and AI-based methods to extract different kinds of morphological information from alternate wing morphology. So like the kinds of data that I get is, there are two kinds of data. The first is the landmark. So there are quite a lot of landmark talk in this workshop, so I don't really have to explain much. But the strength of landmark data is that it is relatively homologizable. So like one landmark from one species or one individual can readily be, comparable with the same landmark of other individuals. So that is a very big, benefit of landmark. And the other trait, which I and Seth actually are getting more and more excited about, is the kind of like a lower level phenotype, this small cell-like tissue that kind of stuck together to form a wing. We call it like a vein domain, and we are basically now using a very simple segmentation method to quantify the size, perimeter, or like thickness of those veins, and identify each location of each of those domains in a wing. And so far, I have digitized about 12,000 wings from 2 species. And there is like maybe 20,000 more wings for me to scan, which I probably should do. And Eric is kicking my ass every now and then, and I should do that soon. But anyway, the data is going to be there. And what we want to do with those is essentially to kind of identify some key features of those phenomic data that will likely to be novel for our ability to study wings. And one of the things is the study of a very rare variant. And here, this is how a normal wing looked like. And these two individuals has a very strange, features of their wing morphology. And those are very rare. It is about 1 to 3% of the entire, individual has this. But now, because we have just 12,000 images, we have about 500 cases of this. And with this, kind of data, we can study how those, unique and rare aberration, like developmental defects, actually could inform us something about the development of those wings. Another interesting thing that we are trying to do is to kind of do a phenotype map. So like we have 2 levels of phenotypes, right? So on the one hand, we have like wing size, wing length, or the global shape. That is kind of the global entity that a wing has as a whole. But we also have like underlying feature, like a domain level phenotype. And the question that we are asking here is very simply, what does it mean to have a long or short wing? Is that because of the areas of each domains are getting bigger, or is there many more domains? And what I seem to find, even though I'm not so confident about this result yet, but what this result seems to tell me is that when the wing becomes larger or smaller or different shape, it seems to be primarily because of the size of each domain seems to be changing, even though, which is quite interesting. And the final thing is to kind of study the subtle variation. And I just want to very quick, is like what we are trying to do here is that we are merging or the matching a pair of wing in terms of their contour as much as possible, and we scale those in terms of their contour. And then we still have slight difference in those pairs of wings, and we can evaluate how a pair of wing is different in terms of the domain pattern, and we color code as the lighter color when there is a little variation, and if there is a high variation. The darker color when there is a high variation. And what it seems is that if you try to flip the left and right side of the wing, there seems to be a lot of difference between left and right side of the same individual, but it seems to have a particular pattern where this dorsal edge seems to have more different in terms of their left-right asymmetry, whereas, and also the variation within a population across different individuals seems to also recapitulate this kind of same pattern where this part of the variation is greater than this part of the variation. And this kind of subtle variation and also the visualization is a very strong feature of phenomic data. So to conclude, what I'd like to say is that, well, the future of phenomics is already bright, but I think it is going to be brighter if we actually are aware of the limitation of the methods. And I think it's a strategic a choice to focus on the most unique feature of phenomics might actually is a good idea to kind of speed up the development of the field. With that, I would like to thank my collaborator, and I would like to thank you for listening. Thanks.